Hello and welcome to Talking Pharmaceuticals, an interview series brought to you by Making Pharmaceuticals and Distributing Pharmaceuticals. I'm Victoria Emerton, Conference Director for the events, which next take place in April 2025. And in the lead up, I'll be joined by experts from UK Pharma to share insights, opinions and practical advice, providing a fresh perspective on key challenges and opportunities in the industry and to look ahead to their involvement in the events. I'm delighted to be joined today by Dr. Michael Black, Head of Pharma Sales from Beneo Palatinate. Michael, thank you very much for your time today.
Thank you, Victoria. I'm really happy to be here.
And we're excited to have you coming to Making Pharmaceuticals in Coventry again. You've been several times, so great to have you involved in the event another time. You are presenting this year again, so we're going to talk a little bit about what you're covering in your presentation, so people know what to expect and can come and listen further and ask you more questions in Coventry in April. So first off, what is lyophilization?
Yeah, so I'm going to talk about lyophilization. It's also known as freeze drying, and it's a dehydration process to preserve molecules, materials, which can make them more convenient for transport and also give them a higher stability. So it generally involves like 3 steps. One is the freezing step. So you're turning the water in the product into ice. And then there's a primary drying step, which is a sublimation step. So the ice gets converted to gaseous water. And then there's a secondary drying step where you remove the remaining water from the substance.
And how is lyophilization used in pharmaceutical technology?
Yeah, well, it's becoming more and more relevant because of the move now towards larger molecules, larger biological molecules. So it's used to preserve and stabilize these, and also looking at vaccines and drugs that are natural products.
So what are the benefits of freeze drying?
Yeah, well, for a lot of materials like proteins, enzymes, vaccines, which are sensitive to heat and moisture, the lyophilization enables to maintain their stability and efficacy by removing the water without exposing them to high temperatures. And secondly, you can then extend the shelf life of this material and also enhance its solubility. So for instance, if you have an injectable material in a powder form, if it's then already lyophilized, freeze-dried, then you can reconstitute it with water much more effectively and quickly, which is then in the practical situation very useful.
And I imagine it's cheaper to transport as well. If you remove water, there'll be less weight involved too.
Exactly, and this is always the question, why do we transport water from A to B? And here we don't need to do it.
Fantastic. So what equipment is required? Is it fantastically complicated?
Well, I've seen sort of lab scale ones which look quite simple, but basically you need to have a refrigeration system, obviously, to cool the material. Then you need an ice condenser. You need a vacuum pump because that's how you remove the frozen water. You need a drying chamber and, of course, a control system fixed onto all of that in order to be able to manage the whole system effectively.
So what's the focus of the study that you're going to present at Making Pharmaceuticals?
Yeah. So this is a study which the title of the study is Physical Stability. The study that we based my talk on is called Physical Stability of Freeze-Dried Isomalt diastereomer mixtures. So isomalt is an excipient which is used in solid dosage forms in the pharmaceutical industry. And the purpose of this study is to determine if it can be a suitable excipient in a freeze drying process. And then looking at the freeze-dried material under different conditions. So that's basically the idea behind the study.
Okay, and anything that you're affecting the physical form of, I guess, in pharmaceuticals, you need to test whether what you're doing to it has affected it in any way. So what analytical methods did you use in this study?
Yeah, so there are quite a lot of analytical methods that are used, studying, for instance, the crystallization tendency of the freeze-dried isomalt. So the freeze-dried isomalt is amorphous, and therefore you have to measure its, let's say, if it's recrystallizing or staying in the amorphous form. So things like differential scanning, calorimetry, X-ray diffraction, infrared spectroscopy, and Raman spectroscopy. All of these methods are used in the study. Also, principal component analysis, PCA, which is used to determine if there are differences between the raw materials and their stability over time. And then a Carl Fisher analysis to measure the water content, and also thermogravimetric analysis also to measure water content.
Fantastic. And you'll be going into a great level of detail during your presentation at the event, I'm sure. So Michael's presentation title is The Physicochemical Properties of Amorphous Isomalt as a Suitable Excipient for Freeze-Drying Formulations. So we'll look forward to hearing that presentation in
Well, I've got a bit of
a
spoiler, Victoria. The study does say that it is suitable. Yeah. So,
okay.
So it's, I know it would have been a cliffhanger, but the study does say that it is suitable for freeze-dried operations. So I'd be really looking forward to people coming and seeing that and asking me some nice questions.
Well, you heard it here first, folks, and hopefully people armed with that information, having watched this, can come and ask you sort of detailed application questions. Now we know it's all good to go. How could they use it? How could it help them? That's the next step, isn't it?
Exactly.
Fantastic. So you're going to be available, obviously, both after your presentation, but you're going to be on the Harker Pharma stand, I understand. So if anyone wants to come along and talk to you before, during, after your presentation, that's where you'll be, which is stand 509. So just to remind everybody that attending the exhibition and the conference is free, and registration is open at www.makingpharma.com. The conference programme is also live. So we look forward to seeing you, Michael, and your colleagues at the Coventry Building Society Arena on the 29th and 30th of April. Thank you very much for your time.
Thank you very much, and I'm really looking forward to the event as well.
Thank you.